Hello friends and viewers. Today I am going to talk about that how we can visualize the immune cell in the laboratory, because we are working in the laboratory and we know that how this cell looks like in the microscope and in the flow cytometry, but most of the people do not have access these machines and they do not know how actually they can be seen in the laboratory. So this is a general mystery to those people that who cannot see the lymphocyte or the granulocyte, how they look like. So to visualize the cells, especially the immune cells, there are multiple techniques, but in this video I am going to talk about the two techniques depending on the microscopy and the flow cytometry. Okay, the microscopy. If you take the blood sample from the venous of any people, and if you just take under the microscope, then you can see a lots of cell, including the RBC and the lymphocyte. But in that stage, it is very difficult to see the immunological cell, especially the lymphocyte, granulocyte, monocyte. So we normally lyse the RBC or red blood cell with some lysing solution, which specifically brushed out the red blood cell, because the red blood cell do not have the nucleus, so very sensitive to lysis. And then we stain rest of the cell with the Type-N blue. Type-N blue is a dye, it looks blue, and this is a dye that can differentiate between the dead cell and the live cell, and also the live cell becomes more visible and very bright under the microscope. So when you prepare in this way your slide and your cell are placed under the microscope here, then you can see a lots of cell on the slide, and those cells are basically the leukocyte, and the leukocyte comprises the lymphocyte, monocyte, eosinophil, basophil and neutrophil, all of the immunological cell. But since the lymphocyte and the neutrophil are the most abundant cell in the blood, you will mostly see the lymphocyte and neutrophil. In higher magnification, you can differentiate the lymphocyte and the monocyte, eosinophil, basophil, neutrophil clearly, but in normal magnification, this cell is little bit difficult to distinguish. And also it depends on the source of the lymphocyte, because the human lymphocytes are bigger than the mice lymphocyte. So if you take the mice lymphocyte, then it is difficult to see than the human lymphocyte. But you can do this depending on their morphological change, size, nuclear shape and the frequency in blood. Normally the lymphocyte have a bigger nucleus and the small size of the cell. Monocyte have a bigger size of cell. Eosinophil has the granule and the bilobed of the nucleus. Basophil have the almost same type of structure, and the neutrophil have a multiple lobe on their nucleus with the same size of the structure, but they have the higher frequency in the blood, so you can easily distinguish depending on this criteria. So in this way, in the laboratory, we see the immunological cell under the microscope, and this is very easy and simple way to see the cells under the microscope. So next I am going to talk about the flow cytometry. Flow cytometry is a more sophisticated technique than the normal light microscopy. So in the flow cytometry, you have the ability to detect very specific type of cell, not only the lymphocyte or the granulocyte. You can differentiate different types of lymphocyte, like the T cell, B cell, CD4 T lymphocyte, CD8 T lymphocyte. So how this flow cytometry actually work? So we have different type of cell surface marker depending on the cell type. For example, the stem cell express the CD34 and the all leukocyte express the CD45 protein on their cell surface. And a specific cell have a specific marker, like the granulocyte have CD15, monocyte have the CD14, T lymphocyte have the CD3, B lymphocyte have the CD19 and thrombocyte CD61. Also T lymphocyte, which are actually helper T cell, they express also CD4, and cytotoxic T cell express CD8, and activated T cell express CD25. And in this way, literally all of the cell in the body have a specific marker on their cell surface. These markers are actually protein that expressed on the cell surface. And if you go online, the BD or Becton Dickinson and the eBioscience, they have a specific chart that show that which cell surface marker is actually expressed on which type of cell. They have also differentiated the human and the mice chart that can easily distinguish the human and the mice lymphocyte with the different types of cells. So we have a specific marker on cell surface, and in flow cytometry there is a specific type of monoclonal antibody used that is specific for this cell surface protein, and those monoclonal antibody is always tagged with specific fluorochrome, and fluorochrome have different color like the blue, red or any color. For simplicity, I am telling that okay, they are the blue or red or the green, this type of color. So when this fluorochrome tagged antibody are incubated with this type of lymphocyte or any leukocyte, they bind to this surface protein. So when you take the blood from the venous and lysis of the red blood cell and incubated those fluorochrome tagged antibody to this leukocyte, and definitely you need to wash out the extra fluorochrome tagged antibody in the solution, and then you run the sample in the flow cytometry machine. Then you can visualize the cell number and the type of cell, and not only you can visualize the specific cell type, but also you can sort a specific cell type. Like if you want to sort out the CD3 or the T lymphocyte, you can sort and pull out those type of cell from total leukocyte. So in this way, the flow cytometry is a very good technique for the immunological purpose that can solve the problem that how you can visualize the cell and how you can sort or pull out a specific type of cell from the total leukocyte in the blood. But how these cells looks like in the flow cytometry? They do not look like in the microscopy. They are not visualized as a structural cell. They are only visualized as a processed cell in the flow cytometry machine. They are visualized as a dot in the screen. Here in the left side, this is the flow cytometry machine, and normally when you run the sample, this flow cytometry machine takes the sample and only one by one cell flow in this machine, and here there is a specific laser that can hit those tagged fluorochrome, and those tagged fluorochrome, when they are heated by specific laser, they emit a specific wavelength of the color. Those wavelength of the color are detected by this machine. For example, if you tag the CD3 marker with the anti-CD3 monoclonal antibody tagged with fluorochrome, like the APC, this is a specific fluorochrome, and the anti-CD19 monoclonal antibody tagged with the PE-Cy7, then this machine can differentiate that APC fluorochrome and the PE-Cy7 depending on their emission of color. Basically, the flow cytometry machine will give you two types of plot. This is the type 1 plot. Here there is no color is detected. Only cells are separated depending on their size and the granularity. The FSC, which is called the forward scattering light, and the SSC, which is actually side scattering light, these two light, after heated by the laser, they are detected by this machine. And if the cell is bigger in size, the forward scattering light put your cell in the right side of this plot, in the x-axis. And if the cell is more granular, then your machine will put your cell in the up side of the y-axis. For example, here is a typical plot of the leukocyte, where you can see these are the lymphocytes which are smaller than the monocyte and the neutrophil. That's why they are left side of the FSC x-axis. But the monocyte and the neutrophil is bigger, so they are right side of the x-axis. And also the neutrophil, which is more granular than the lymphocyte, they are positioned in the upside of the y-axis. In this way, the flow cytometry machine can tell you that which cell type are where position, and you can literally count this cell, because here every dot is a one cell. So in this software of the machine, you can get this lymphocyte, and this machine will tell that how many lymphocyte in there, and you can get also the monocyte or neutrophil, and then the machine can tell you that how many neutrophil, monocyte and the lymphocyte in the sample. And the second type of plot, which is actually depending on the fluorochrome tagged. Here you can see the CD3 and the CD19 are tagged with a defined fluorochrome, like the CD3, which are actually T lymphocyte, tagged with the APC, and the CD19, which is tagged with the PE-Cy7 fluorochrome, are the B cell. So when you found the first plot, you can get this lymphocyte, and from this gate you can separate the T lymphocyte or B cells depending on the fluorochrome. And in this software, the software will give the total count of the B cell and the T cell from this plot. So this is how we actually count the cells. And later, if you give the command of this machine to separate of your these gated B lymphocytes, then this machine actually sort out those lymphocyte in a specific tube or a plate. So in this way, we in the laboratory detect and see and visualize the immunological cell, and sometimes we sort and pull out the cell from the total leukocyte for our experiment. Hope you have enjoyed the visualization of the immunological cell in the laboratory. Thank you for watching. If you think that this is very easy to understand and very helpful for you, please subscribe, like and share.